in general, prokaryotic cells have a very, very simple form. So we don't have a lot of physical diversity between types of bacteria or types of archaea. There are some specific structural differences in the membrane and in the cell wall between bacteria and archaea, but overall, their general structure is going to be very, very similar. They're single-celled organisms or very, very, very simple multicellular organisms. And they have just one cell with no organelles. So in all of our prokaryotic cells, we're basically going to have a membrane, a cell wall, and the DNA inside. So there's very little morphological differences between prokaryotes. They do come in 5 major shapes. Generally, they are spheres, rods, and spirals. Now you'll notice here there are a couple other variations. The sphere-shaped bacteria are called cocci. There's an example here. These are basically just round sphere-shaped bacteria. We also have rod shaped. So these are bacteria that are long and thin. And we've got an example here of those guys. The next general structure is a spiral structure. So these are usually called spirilla or spirillum for the singular. And you can see here we've got these ones that are just very vaguely spiraled, and we've got these ones that are very, very twisted and spiral. So some of them are rigid, some of them are flexible. In general, we're just going to kind of distinguish them as sphere, rods, and spiral. Those are the 3 shapes that you'll need to be able to identify in your lab. We're going to look at those specific shapes. So you do need to know those general shapes. You also need to know the microbiology term for that. Spheres are cocci, rods are bacilli, and then our spiral ones are usually called spirillae or spirillum, but we do have these specific groups called the vibrios and the spirochetes. So generally, if you call something a spirochete or a spirillum, that's going to be perfectly fine. The vibrios are a specific group of organisms, and so it's a little more restrictive than the general spiral-shaped. So the general structure of our bacteria is shown here. So whether they are cocci, bacilli, or spirilla, they are going to have all of these general characteristics. So inside our bacterial cell, we have the nucleoid region. So this is a region filled with DNA. It's kind of like a concentrated area of DNA. Now notice that there is no membrane around that DNA, because if there was a membrane, it would be a nucleus and we would have a eukaryote. Now, there are a couple of examples of bacteria that do actually have a membrane around their nucleoid region. This is not considered a nucleus. It's a different type of membrane. So if you happen to Google, like, do prokaryotes have nuclei? There are like 1 or 2 weird examples where they do have membrane separated DNA, but it's still not considered a true nucleus. All of our cells have ribosomes, so whether you are eukaryote or prokaryote, you have ribosomes. This is where we're going to produce our proteins. So those are not unique to prokaryotic cells. Around the cell itself, surrounding the DNA and the cytoplasm, we've got the plasma membrane. For bacteria, this is going to be made up of phospholipids. Around that, we have a cell wall. So this is going to be a rigid structure that's going to give the bacteria support in its environment. It's going to give it kind of a 3-dimensional shape and a 3-dimensional structure. On the outside, we have a capsule. This is where you're going to find the mucilage that I talked about a couple of times. We'll talk about that in a minute. The capsule is kind of an outside covering. Sometimes it's kind of slimy and sticky. and it's just another protective layer around the bacteria. They also may have a pilus. So we talked about a specialized form of a pilus called the sex pilus. This is used in conjugation. Bacteria may also have pili that help it move, kind of like cilia, if you remember cilia from eukaryotic cells in biology 101. But the pilus is usually has some sort of specific purpose, not necessarily related to movement, although it can be. And then many have a flagellum. This is to help with movement. So if you think about sperm cells, so sperm cells are eukaryotes. They are not bacteria, but they have these flagellum as well. And their purpose is to help them move through their environment. So they're kind of interesting. They have this little protein motor structure here that helps them beat and helps them move around. So this is a way for the bacteria to move inside their environment. So bacterial cells have cell walls. They do vary in structure just a little bit. Archaea have cell walls as well. Their which we find in plant cells. It's basically repeating chains of glucose that are sort of all interlinked to sort of form like basically what would be like an interwoven mat of glucose molecules. And it's really there to give that cell a rigid outside structure. So this is not the same origin as plant cells. Plant cells evolved later. This is a very different structure, but it does the same purpose, which is to give those bacteria a rigid structure. So it helps the cell to maintain shape. It helps to protect against attack. It helps to protect against infection, so against viruses, against conjugation and whatnot. It's not perfect. We do still have viral infections. We do still have conjugation happening, but it does help to protect against that. It also helps the cell to avoid lysis. So lysis means to break. And a cell might do this when it is in a hypotonic situation. So I'm not super worried if you don't remember what this means, but in a hypotonic solution, basically what's going to happen is water is going to rush in to the cell. So this depends on the concentration of the cell. But what happens here is if there's a particular gradient in concentration of solutes inside and out, water can actually rush in. Now, without a cell wall, that cell is going to lyse and break. And so the cell wall kind of pushes back so that the cell membrane cannot break if water rushes in. This is really important for bacteria. This is also important for protists, although they have a different mechanism for dealing with this. So we have two major types of bacterial cell walls, and they are named after a staining process. So in lab, we are going to do something called a gram stain, and this is a really quick way to determine one of two very broad types of bacteria. So the gram stain is going to distinguish between what we call gram-positive and gram-negative bacteria. Now, this has to do with how the stain sticks to the cell wall. So there are two different types of cell walls. The first type is what we would call a gram-positive stain. And this stain sticks to the bacteria because they have a really, really thick layer of peptidoglycan. So that's the the molecule that makes up the cell wall. This image over here on the left, we've got the plasma membrane. So there's the membrane here. And then we've got this really thick cell wall structure. So you can see this is kind of like an interwoven mat. That's what it was about. It's really, really thick layer of these peptidoglycan molecules. And you'll notice that it's on the outside of the plasma membrane. So that's an important characteristic of these gram-positive bacteria. So we call these gram-positive because when we do this gram stain process, that thick wall maintains the stain. So it holds that gram stain. Now it does that because it's that really thick wall. It sort of gets integrated into the wall of the bacterial cell. So this particular cell wall is actually vulnerable to penicillin. So penicillin is an antibiotic that kills bacterial cells, and it works by interfering with cell wall synthesis. So if the bacteria can't establish their cell wall, then they're not going to survive. During our lab, for this particular chapter, you are going to do this gram stain, and you are going to identify whether your bacteria are gram-positive with this really thick peptidoglycan wall, or if they are gram-negative. So gram-negative is the other option. The overall structure for these guys is just a little bit different. So on gram-negative bacteria, you have the plasma membrane, and then you have a peptidoglycan wall on the outside. Now you'll notice that compared to this one up here, this wall is much, much thinner. And then you've also got this outer envelope. So this is kind of a secondary plasma membrane that occurs outside of that cell wall. So the gram stain doesn't stain as strongly onto this thinner peptidoglycan wall. So basically what happens is you get some stain, but not as much stain as you saw in this gram-positive bacteria. So those thinner walls lose their purple stain, but they do retain a little bit, and so they look this kind of bright pink color. These guys resist penicillin because they have this lipopolysaccharide layer, this outer envelope, this sort of secondary plasma membrane, and that actually blocks the penicillin from getting to that cell wall. So these guys are not susceptible to penicillin, and they require other antibiotics instead. So the cell wall structure is a major defining feature for establishing what type of bacteria you have. So I've mentioned the mucilage just a couple of times. This comes by a couple of different names depending on the textbook you're looking at. Most often, if it's not called a mucilage, it's going to be called a capsule or a glycocalyx. And again, this is just the structure that is on the outside of the membrane and the cell wall. So it has a few different functions. Basically, it is kind of used for protection, kind of an overall function of protection, but there are some specific ways that it's used. So mucilage is basically a slimy coating, so think of the word mucus. That's what this is. It's basically just mucus on the outside of the cell, and it's going to help in defending against invasion by viruses, conjugation. It's also going to protect against UV damage. So just like UV rays can damage our cells, they can damage pretty much any living organism, because UV is going to interrupt the bonds between DNA nucleotides, so the bonds within DNA. And so we don't want that. So it's just a sort of protection, kind of like a sunscreen against UV damage. They also work to hold colonies together. So we talked about those cyanobacteria mats. Those colonies may also be called biofilms. So this is kind of a thin layer of cells along a surface. Dental plaque is actually an example of a biofilm. So if you are not removing that plaque, it turns into that hard tartar buildup. So when you get a whole bunch of bacteria together in a biofilm, it can actually change structure, and this is why we get things like stromatolites and easy fossilization of these mat-like bacteria. So we mentioned bacteria motility. In general, bacteria are moving through their environment with this flagella structure. So the flagella can allow the bacteria to swim. It kind of twitches, glides, or even just kind of floats through the environment. But the flagella can help with that movement. So I mentioned earlier there's this sort of protein motor attached to this long filament that basically kind of moves back and forth and twitches, and it's going to help that propel through the environment. This is important because this is one of the ways that bacteria can move through your body and spread infection. Your textbook does have a great description of how the motor protein works. We're not going to go into that, but if you're interested, it is there in your textbook. So last little bit, there's a huge diversity in bacterial nutrition. The two major groups are going to be our autotrophs and heterotrophs. So autotrophs can produce their own organic compounds from inorganic ones. So our sort of common example of this are our photoautotrophs. These are the ones that perform photosynthesis. And they are going to take inorganic carbon in the form of carbon dioxide, and they're going to convert it into glucose. Now, there are other examples, other types of autotrophs. That we have organisms that can use sulfur, we have organisms that can use nitrogen, other chemicals, inorganic compounds that can be converted into organic energy storage molecules. We also have heterotrophs. So these are organisms that cannot build their own organic compounds. So instead, they have to get their organic molecules from the environment. In other words, they have to consume them. They have to consume their organic molecules. So you and I are heterotrophs. Many of our eukaryotes are heterotrophs. Basically, for eukaryotes, if they're not performing photosynthesis, they're probably heterotrophs. But bacteria and archaea can be either one. They can either, some groups will act as autotrophs, others will act as heterotrophs. Continuing on with our bacteria metabolism, the way that they utilize oxygen is also really important. So we have a couple of different terms here. We have got the term aerobe. These are organisms that use oxygen. So the oxygen is usually required in order for them to survive. So we are aerobic organisms. Early in life history, we talked about how there was very little oxygen on early Earth, and so most of the early organisms were probably anaerobes, which did not utilize oxygen. And then aerobic organisms came later when we had cyanobacteria releasing more oxygen into the environment. We have several categories of anaerobes. So these are organisms that either can't or don't use oxygen. So they come in a couple of different levels. So we've got obligate anaerobes. These guys cannot even live in the presence of oxygen. So oxygen's going to kill them. So they are utilizing some other source, usually sulfur, nitrogen, or methane, as their inorganic molecule that they're using to take in energy. We also have aerotolerant. So these guys don't use oxygen, but they're not poisoned by it either. So yeast are an example. Yeast are actually fungi. That was a bad example. It just popped into my head. But they are an example of an aerotolerant anaerobe. They don't use oxygen, but they're not poisoned by it either. So usually these guys, bacteria or fungi, whatever happens to be performing them, are performing fermentation, so alcohol fermentation or lactic acid fermentation, or a form of anaerobic respiration. And then we also have facultative anaerobes. They can actually switch between using oxygen or not, depending on the conditions. So yeast are actually, or some yeast can actually be facultative anaerobes. They can actually switch between using or not using oxygen, depending on whether or not oxygen's available. So we mentioned nitrogen fixers a little bit ago. Cyanobacteria and many others are important in fixing nitrogen gas. This basically means that they are taking inorganic nitrogen and converting it to an organic form that is going to be required for algae and plants. These guys are autotrophs, so they're not eating other organisms, and so they need nitrogen to come in so that they can build their organic molecules that require those molecules. So example of that would be amino acids. Our nucleic acids also require nitrogen. And so in order to be able to have nitrogen in their cells, they have to get it into an organic form. So one of the key functions of cyanobacteria and many other prokaryotes is to actually convert that nitrogen gas into a usable form.